Hi everyone, it's me, Dr. Suresh Kumar, biology expert for the competitive examinations and regular board examinations also. As I discussed previously, the next part is nothing but part 5th of the sexual reproduction in flowering plant. Let us discuss in this part what is present, what I would like to discuss to you. Now, lesson 1, sexual reproduction in flowering plant, part 5. What is the subtopic of this part 5th? What I would like to discuss to you mean? Androecium, stamen, microsporangium and pollen grain. All these are belonging to the male reproductive part. Now here, before talking about the descriptive level and objectives, we need to have some sort of schematic representation. See here, this is the bisexual flower. Here, 4 parts are present. One is the calyx, corolla, androecium and gynoecium. Now here we are talking about the only male reproductive system. In the male reproductive system, what is there means only the stamen we need to talk. Here in the stamen, exclusively 2 parts are present. One is anther, second one is the filament. Now here, in the anther point of view, inside pollen grains, how they are developing, we need to come to know that. The transverse section of the anther is having a tetragonal shape. Each end of the angle, there is one kind of sporangium. So, each anther is having the 4 sporangia. Each sporangia is consisting the number of pollen grains. So, this is the schematic representation we need to understand. It's the diagrammatic representation. Then next, if you go for same thing is given in the schematic representation in the word formation. Androecium, stamen, anther, microsporangium and pollen. Now here we can come to know that androecium is composed with the stamens. Stamens are consisting the anther. Anther consists the microsporangium. Microsporangium consists the pollen grains. Now here, what is meant by androecium? So if you come to the point here, what is the androecium means? First of all, what kind of organ we need to talk? It is a male reproductive organ. As you know that the flower is having the 4 parts. 2 are non-essential, 2 are essential. Here what are the non-essential mean? Calyx and corolla. And what are the essential means? Androecium and gynoecium. Here if you see that the male reproductive organ is nothing but androecium. Then a collective form, if you see that, a group of stamens collectively known as also androecium. These 2 meanings will come for the androecium. Now, you see what is the structure of stamen? See, stamen, this is the diagrammatic representation. You can see this is the one blue color is there, that is the anther, and this color is nothing but elongated filament. So each stamen is 2 collectively called as stamen. So here 2 parts are present. That is the descriptive manner we have mentioned here. Each stamen consists of 2 parts. One is filament, second one is the anther. Now here we need to discuss few points regarding the filament, at the same time few points regarding the anther. Now, what do you mean by filament? A long, this is only, here this structure we are describing here. A long and slender part of the stamen is known as filament. Now, this is the blue color, is the long and slender part of the stamen. Now here if you see, it has 2 ends. This is one end. This is one end, at the same time this is another end. So this end is very close to the thalamus where the floral parts are attached or formed. So this is called as proximal end, and the far from this thalamus the end is called as terminal end. Now here we need to talk about the filament attachment. Based on the filament attachment, the flowers, the stamens are different types. So here if you see, the filament is attached to the thalamus. Hence it is called as epithalamus. Epithalamus means that filament is present on the thalamus. So if the stamens are present on the thalamus, those it is called as epithalamic stamens. Then sometimes what happens, there is a colorful parts in the flower is nothing but a petal. Sometimes some of the filaments are attached to the petal. Then such kind of petals are known as epipetalous. Epithalamus and epipetalous. These two we should remember. So we will ask according to epipetalous means the stamens present on the petal. Then it is called as epipetalous. Next point. The number and length of the stamens. If you see that there are different kinds of species of flowers are there, plants are there. All these plants are not showing the same size and same shape of the length of these stamens. So the number as well as length of the stamen will be showing the variation from one species to other species. It means in plant kingdom, in all flowering plants, the stamen's length and stamen's shape is not look like same. So here we can easily observe different shapes and sizes of anther of different species of flowers by using one kind of microscope. There are different kinds of microscopes are there. How can you identify the shapes and sizes of this filaments? That means immediately we can go for dissecting microscope. This is a neat examination question for us. Dissecting microscope. Then structure of, next one if you see that, structure of angiosperm anther. See that the anther internal structure. See here this anther, bilobe anther we are taking. Then we are taking the transverse section. Under the transverse section we can see like this. Now here you see this is a one lobe. Each anther is having the one lobe like this. This is a one lobe, this is a second lobe. Then each lobe is having the two theca. So here the angiosperm anther is bilobe tetrathecus. Tetrathecus 1, 2, 3, 4. Now we would like to talk about all these things. First, a typical angiosperm anther is directly we can say that it is a bilobe. Bilobe, two lobes are present. Next, each lobe is having two theca. Each lobe is having two theca like this. This is one lobe. This lobe 1 and 2. Two theca are present, 1 and 2. Here second lobe if you see, third and fourth theca are present here. So basically the angiosperm anther is two lobes are present. That's why it is a bilobe. Four theca are present. Hence it is called as tetrathecus. So here each lobe is having the two theca, hence it is a dithecus. How one lobe of the anther is divided into two theca? You see that this is a one lobe. It is some kind of tissue is traveled from the longitudinally and form the two theca, 1 and 2. So here you see that one theca is divided into, one lobe is divided into two theca. Now here answer for the above question. How one lobe of the anther divided into two theca? Now here answer is present. A longitudinal groove runs lengthwise of each lobe and divided one lobe into two theca. So in a bilobed anther there are total 4 theca are located. Total 4 theca are located. Now, if one anther, this is application question based on the previous statement. If one anther has 4 lobes, previously 2 lobes only studied. Now I am talking about the 4 lobes. If one anther has 4 lobes, and each lobe is divided by a longitudinal groove. Then how many theca present in it? How many theca it has? How many theca it has? Now here you know that one lobe is having 2 theca, then 4 lobes it is having 4 lobes, 4 into 2 it is nothing but 8 theca. This is an application question for us. Next, afterwards, the transverse section is commonly called as TS of bilobed anther, that is having the shape tetragonal in shape, that means 4-sided. That shape is like this. This is a bilobe here as previously I mentioned. This is a one lobe, this is one lobe, and this is the second lobe. In the second lobe it is having 4 theca, 1, 2, 3, 4. Then here the tetragonal shaped bilobed anther consists of how many microsporangia? How many microsporangia? 4 microsporangia. That is also complete question for us. Next, each microsporangium at corner of tetragonal anther, each microsporangium is present at the corner of the tetragonal anther. Location they will ask the question to you. Next, afterwards, each microsporangium further develops into a pollen sac. See here the pollen sac further. This is a microsporangium. After the microsporangium, if you see that, if you take out this microsporangium, inside some sort of pollens are developing. Then here each microsporangium developing a pollen in its pollen sac. Now, pollen sac. Finally, pollen sac. Finally, a bilobed anther consists 4 pollen sacs. This is also a new question for us. Next, later each pollen sac extends longitudinally throughout the length of anther. This is also one kind of line in the NCERT. Now, each pollen sac packed with the number of pollen grains. So we need to understand all these things very clearly. Then further continuation in the next part. Now, as I am showing to you, slowly scrolling off the screen, just go ahead. Here you need to focus epithalamus, epipetalis. Here also what kind of microscope we are using, the identification of the shape of the stamen or else the shape of the anther and the structure or shape size of the anther. Dissecting microscope. Bilobed tetragonal. Wherever the blocks are given, those are the important questions for the descriptive as well as the important questions for the NEET examination, competitive examination. Please notify that. This is the application question has given. Please go through this. Thank you all. Please go through. If further any questions are there, please give one question comment and I will follow to you. Thank you all. All the best.